Hello, attendees of Meeting on the Mesa. I'm very pleased to have this opportunity to speak with you about Century Therapeutics today, and would like to thank ARM and the meeting organizers for including Century's presentation. I'm Adrienne Farid, the Chief Operations Officer at Century, a leading iPSC-based cell therapy company where we're focused on researching and developing hematology and oncology treatments. These are my forward-looking statements. At Century, we're building a next-generation allogeneic cell therapy platform. We've built foundational expertise in several core areas, including these four key pillars: iPSC, or induced pluripotent stem cell reprogramming, gene editing, protein engineering, and in-house manufacturing. While we're developing allogeneic cell therapies, we differ from donor-derived cell therapies by utilizing iPSCs as our starting point for NK and T cell therapies. We've started with a comprehensive collection of CD34 positive human stem cell, alpha beta T cell, and gamma delta T cell derived clinical grade iPSC lines. Use of iPSCs versus donor lines is powerful, as I will illustrate. We use a proprietary platform for gene editing, the MAN7 enzyme for homology-directed repair utilizing CRISPR. This is used for precise gene integration. Within our protein engineering area, we have the ability to optimize chimeric antigen receptor binding, signaling, pairing, and we're also gaining insight into a universal tumor targeting platform. Another important key pillar is our in-house and operational manufacturing for iPSC drive products. Our scalable protocols and processes include differentiation to produce highly functional NK and T cell products that can be well characterized. At left is an illustration of our cell editing process to form the engineered iPSC cell lines. Since we're editing induced pluripotent stem cells as opposed to donor-derived NK or T cells, we can make edits in a sequential fashion and the cells continue to replicate and do not exhaust. This also avoids multiplex modification or making several edits at one time, which may lead to undesired structural variants. We're using CRISPR-MAN7. Earlier this year at ASGCT, we had communicated that MAN7 editing fidelity is on par with the gold standard CPF1 enzyme. And importantly, we isolate a single cell clone to form a homogeneous master cell bank with product consistency and integrity. We're able to fully characterize the master cell bank. Manufacturing then begins at this master cell bank, already confirmedly created genetic. So in essence, near unlimited expansion potential of iPSCs in both the genetic engineering and the manufacturing processes. All of Celgene's cell therapies include alloevasion editing, illustrated here. The purpose of alloevasion is to prevent immunological rejection of the cells and enhance persistence in a patient dosing setting. So as you see in the illustration on the left, the genome editing or knockout of first beta-2 microglobulin B2M and second C2TA, which is a protein regulating MHC class II transcription, those knockouts respectively disrupt the expression of HLA class I and HLA class II on the cells. So these two cell edits prevent recognition of our cells by CD8 and CD4 T cells. Moreover, we've utilized knock-in of HLA-E and HLA-G to prevent NK-mediated lysis of our cells. The net result of alloevasion edits is to avoid major pathways opposed versus graft rejection, all of the major pathways, and thus prevent immunological rejection of the cells and enhance persistence of the cells in vivo. Importantly, these features should allow for multiple dosing regimens with the aim to increase the proportion of patients that achieve durable responses by allowing them access to multiple dosing regimens. Recently, we have achieved a major milestone within our iNK common progenitor cell line that leverages all of the expertise and specialization in the cross-functional areas previously discussed. So production of this common progenitor, which is an iPSC bank with 12 port edits introduced in 5 sequential steps, is allowing us to move more quickly and more avoid some of the risks toward development candidate selection. The specific functions are advantageous. We can include specific functions advantageous for certain cancers, such as our lymphoma and glioblastoma product candidates. Different CARs or different specific edits can be further incorporated prior to manufacturing and differentiation to NK cells. So this common progenitor bank then enables rapid generation of new candidates to realize synergies. I'd like to take a few moments to walk you through our pipeline. Century 101 in the top row is our CD19 directed elevation-enabled B-cell malignancy product, scheduled to enter the clinic later this year. Our IND was submitted mid-year and we received safe procedure for the Century 101 phase 1 trial named ELIPS-1. We're really highly committed to make progress in B-cell malignancies, since despite the high competition in the relapse refractory B-cell malignancy space, there's still a large unmet medical need, and we're working on follow-on products as shown, including Century 102. Century 103 is our GBM targeted product candidate. This CD133 directed product is based on the iNK common progenitor version 3. Pictured earlier, and I'll say a little more on subsequent slides. We also are working on Century 104 and 106 under a discovery collaboration with Bristol Myers Squibb. So we've partnered programs in the 2 different indications, AML and multiple myeloma, both intended to be multi-specific, either IT or iNK candidates, and they should address specific challenges in these disease areas. We're excited to work with BMS, who brings extensive hem-onc experience in multiple complementary technologies, and the collaboration is proceeding well. We also are working on additional research programs in both solid tumors, mainly focused on the gamma delta IT cell platform, and in heme diseases. A little more about Century 101 is shown here. As mentioned, this is our CD19 directed elevation-enabled product. Century 1 includes beta-2M and C2T knockout, and also HLA-E knock-in, as described earlier, designed to avoid major pathways of rejection. In addition, Century 101 includes cytokine support, secreted IL-15, which enhances persistence and functionality of the cell, and a mechanism for elimination, the safety switch, that's a short EGFR. We're pleased to report that we've received safe to proceed notification for the initial phase 1 study named ELIPS-1, which is on track to initiate later this year in patients with relapse refractory CD19 positive B-cell malignancies, including aggressive and indolent lymphomas. ELIPS-1 will assess the safety, tolerability, pharmacokinetics, and preliminary efficacy of Century 101 in patients. The main objectives of the study are to evaluate safety and determine the maximum tolerated dose and regimen of Century 101. The study will evaluate single and multiple dosing regimens of Century 101 with IL-2 following lymphodepletion. And as noted, patients who demonstrate a clinical benefit may be eligible for additional cycles of treatment pending FDA agreement. We're really looking forward to getting insight into these key objectives and to evaluating whether elevation may allow for more effective repeat dosing protocols for patients. A little bit more about Century 103, our first program in CNS malignancies. This is directed at recurrent glioblastoma, which has a poor prognosis, and we're very eager to make some progress in this disease. This CD133 directed INT program is built on the common progenitor version 3, described on the previous slide, and will take advantage of, in addition to the allo-evasion edits mentioned, 3 different tumor cell killing mechanisms. So first, a CD133 chimeric antigen receptor mediating tumor recognition, CD16 mediated killing upon combination with therapeutic antibodies such as anti-EGFR, which kills via ADCC, and also NKG2D mediated innate cell killing, which is relevant in GBM. And a safety switch is also included into this product for cell elimination if ever that's needed. I haven't yet described Century's actively working on gamma delta T cell platform as communicated earlier this year publicly. We have chosen to focus the gamma delta T cell platform on solid tumor treatments, and will communicate out further at a medical meeting later this year, including naming a development candidate in this fall at the R&D Investor Day. Our vision for meeting the barriers observed in cell therapy treatment of solid tumors is shown here. So including allo-evasion, avoiding the major immune recognitions, and cytokine support of the cells to improve functionality and persistence. We're utilizing gamma delta T cell functionality and at least 3 different modes of tumor targeting and tumor killing mechanisms, including tumor antigen binding, ADCC, to enable or including combinations with antibody therapeutics by binding to CD16 and NKG2D recognition of stress ligands. We'll be considering, we're also considering including a safety switch and other functionalities to tackle the specific challenges of the tumor type. Really, as mentioned, later this year, more to come on the gamma delta T cell products in solid tumors. In conclusion, Century is a leading iPSC-derived NK and T cell therapy company. The company has a strong cash position with runway into 2025. Our core technologies and iPSC-derived NK and T cell platforms are progressing very well. Based on the iPSC platform, we have several unique abilities that we believe provide the potential to deliver a homogeneous cell therapy product, manufacture efficiently and consistently to NK and T cell products after we've characterized genomic integrity, enable us to evaluate multiple dosing regimens to see the potential of allo-evasion to increase durable responses to patients, which will be first evaluated via the ELIPS-1 study. We are making strong progress toward the major milestones this year, including phase 1 entry, communication of gamma delta IT preclinical data later this year, and progress on the INK common progenitor version 3 for Century 103 and additional programs. Thank you very much for your time and attention today. I wish you a wonderful meeting.